Hello, welcome to today's class. In this lecture, we're going to carry off where we left off previously, talking about glycogen. This correlates to section 12.8 in your textbook. As a bit of a recap, what is glycogen? Glycogen is a branched polysaccharide composed of glucose molecules. It is used primarily for animal storage, at least within animals. In other words, it stores, glucose. The branched nature of glycogen is particularly important because its structure aids in its function. In particular, the branched nature of glycogen allows for rapid metabolism and or synthesis of glycogen in response to changing environmental conditions. This is because glycogen can only be broken down or consumed from the ends of its glucose chains, shown over here in this schematic. The more ends branched you have, the more ends or terminuses you have to be acted upon by your various glycogen breakdown and glycogen synthesis enzymes. Last class we talked a little bit about the structure of glycogen, a schematic which is shown over here. As a brief overcap, glycogen is formed of links of glucoses. These long linear chains of glucoses are formed by what are called 1,4 glycosidic linkages. In other words, the number 1 carbon of our glucose over here is linked to the number 4 carbon of the subsequent glucose over here and vice versa, giving rise to a 1,4 linkage, as shown over here on the bottom. These 1,4 linkages are responsible for your linear chains. The branches are caused by 1,6 linkages, which, as you can guess, connect the number 1 carbon of one glucose molecule to the number 6 carbon of a second glucose molecule, and this gives rise to our chains. Again, glycogen can only be synthesized or broken down from the 4' position over here. These are called non-reducing ends and are indicated here below. As a final note or reminder, glycogen is used very differently between the various tissues. In the liver, glycogen is broken down to yield glucose 6-phosphate, which can then be converted to glucose via the enzyme glucose 6-phosphatase. In the muscle, however, glycogen can only be broken down to glucose 6-phosphate. The muscle lacks the glucose 6-phosphatase enzyme to convert this back to glucose. What this means is that in the muscle, the glucose stored in glycogen can only be used for glycolysis, since glucose 6-phosphate is impermeable to cell membranes and cannot be exported. In contrast to this, in the liver, glycogen can be used to directly produce glucose. This glucose is membrane permeable and can be exported to other tissues. This reflects the different function of our various tissues. Remember, muscle is primarily concerned with producing energy, whereas the liver has a role in providing fuels for all the other tissues in your body, hence the need for it to be able to produce glucose from glycogen, whereas muscle is not really concerned about this. Long story short, all the glucose stored in the muscle via glycogen can only be used within the muscle and cannot be exported. So now in this class, we're going to talk about the synthesis, or the breakdown and the synthesis of glycogen. First, we're going to start off with glycogen breakdown, or glycogenolysis. As we mentioned earlier, glycogen can only be broken down from the non-reducing or 4' ends. This process is catalyzed by 3 enzymes, which, as you can guess, correspond to 3 different steps or processes. First, we have glycogen phosphorylase. We then have glycogen debranching enzyme, and finally phosphoglucomutase. Note that in this process, we convert glycogen to glucose. We convert glycogen to glucose 6-phosphate, and no energy is required in this process, and we'll see how this goes. Looking at the first step, glycogen phosphorylase, this enzyme breaks the 1,4 glycosidic bond, as shown over here in this schematic. As you can see, we have our glycogen drawn here schematically. Note that our glucose molecules are shown here in a chair conformation. A throwback perhaps to your organic chemistry days where we looked at hexanes. This is a very similar principle. The 1,4 glycosidic bond is shown over here. This bond is broken using a free phosphate molecule to yield a glucose 1-phosphate as well as the glycogen, remaining glycogen chain. The full mechanism for this reaction is shown in the scan notes available in Moodle. Note that this reaction is relatively unfavorable under standard conditions with a delta G of around plus 10 kilojoules per mole. However, in the body, this reaction is driven forwards by the high concentration of free phosphates within the cell. This gives rise to a delta G value of around 3 kilojoules per mole under physiological conditions. And again, the full mechanism for this reaction is shown in our scan notes. One thing which I want you to keep in mind when you look at the reaction is the fact that this reaction occurs via a half-chair cation intermediate. That is its sort of party piece, and again, details of that are available in the scanned notes on Moodle. The second step of glycogen breakdown is the debranching enzyme. The role of the debranching enzyme actually comes from the structure of the glycogen phosphorylase. Shown over here is a schematic illustrating glycogen phosphorylase. This is a relatively chunky enzyme, and the active site is buried somewhere deep inside the enzyme. As a result of this, glycogen phosphorylase has a minimum chain length upon which it can operate, about 4 glucoses. Any shorter, and the glycogen chain cannot actually penetrate far enough into the enzyme to reach the active site. Think of glycogen phosphorylase as a T-Rex with its stubby arms. It has to have a minimum length of glycogen in order for the glycogen to reach its mouth or the active site. Otherwise, it just won't be able to catalyze the reaction. The debranching enzyme basically takes a 4 glucose unit from glycogen and transplants it onto an existing chain to generate chain. This generates a much longer linear chain, which can then be acted on by our glycogen phosphorylase enzyme. Note that our debranching enzyme can cleave 1,4-glycosidic linkages. However, it is unable to cleave this 1,6-glycosidic linkage at the branch point. As a result of debranching enzyme, has to leave a single glucose with its 1,6 linkage. This 1,6 linkage can, however, be hydrolyzed as normal using other enzymes to just give us free glucose. And again, further details of this are available in the scan notes on Moodle. The third step of this reaction is phosphoglucose mutase. This converts the glucose 1-phosphate product from from step 1 into glucose 6-phosphate. This is a relatively simple reaction, and the mechanism is shown over here. This proceeds via a serine phosphate intermediate. So, serine phosphate intermediate. And again, further details are again available in the scan notes. As with before, just to reiterate the point, what I mentioned earlier, is that, just as a reminder, glycogen is used differently depending on your tissue. In the muscle, our glycogen is converted to glucose 6-phosphate. This is the end of the line. We have glucose 6-phosphate, and this is used directly via glycolysis. Whereas in the liver, this glucose 6-phosphate can be either used for glycolysis or can be also converted back to glucose via glucose 6-phosphatase. This enzyme, glucose 6-phosphatase, is not present in the muscle, and thus it cannot do this. So now we've taken a look at the breakdown of glycogen. Let's start looking at the synthesis of glycogen, or the reverse pathway. The relationship between the two processes is shown over here schematically. On the left, we have the breakdown of glycogen via debranching enzyme, glycogen phosphorylase, and then phosphoglucomutase to end up with a glucose 6-phosphate. The synthesis of glycogen happens via an entire separate pathway, kind of mirroring the split or the divide between glycolysis and gluconeogenesis. So to synthesize glycogen, we start off with a glucose 1,6-phosphate. We convert to glucose 1-phosphate. We then have a series of 3 different enzymes to convert our glucose 1-phosphate back towards glycogen. We've got UDP glucose phosphorylase. We have glycogen synthase, and then finally we have branching enzyme. Note that the synthesis of glycogen does require an input of energy in the form of UTP, which is basically the equivalent of ATP except with a uracil rather than an adenine, but the energy content is relatively the same. Shown here is the net reaction for both the synthesis and breakdown of glycogen. As a reminder, glycogen breakdown requires a free phosphate but does not actually require any input of energy, whereas the synthesis of glycogen does require the input of energy via UTP. So we'll go through the various steps. The first step of this reaction is to convert glucose 1-phosphate back to glycogen is UDP glucose phosphorylase. This enzyme is named for the reverse reaction, interestingly enough. In this enzyme, we start off with a glucose 1-phosphate as well as a UTP energy source. Our glucose 1-phosphate carries out a nucleophilic attack on the alpha phosphate, in other words, the phosphate closest to our UTP, shown over here. This results in the production of our UDP glucose, or uracil diphosphate, linked to our glucose molecule. This also results in the release of a pyrophosphate, which is basically 2 phosphates linked together, PPI, or pyrophosphate, shown over here in this sort of orangey color. This pyrophosphate is extremely unstable and quickly hydrolyzes itself to yield 2 free phosphates. It is this hydrolysis reaction of a pyrophosphate into our 2 free phosphates that actually drives our reaction forward, since this reaction is extremely exothermic. We can think about this relationship as follows. Here I've shown the reaction catalyzed by UDP glucose phosphorylase. Here we have a glucose 1-phosphate plus UDP, giving us our UDP glucose plus our pyrophosphate product, shown over here. The delta G for this reaction is actually close to neutral, around 0 kilojoules per mole. What actually provides the energy to push this reaction forwards is the subsequent hydrolysis of our pyrophosphate to yield our 2 free phosphates. This has a delta G of around negative 19 kilojoules per mole, which is sufficiently spicy. If we take the net total of these reactions, we get the following. We have glucose 1-phosphate plus UTP gives us our UDP glucose product plus our 2 free phosphates, and the delta G for this overall net reaction is around negative 19 kilojoules per mole. In other words, this reaction is relatively neutral in terms of thermodynamics. It is the hydrolysis of our pyrophosphate product that ultimately drives this reaction forwards, and is a neat example of a case of how our body can use thermodynamics to drive what are otherwise unfavorable reactions forwards. The second part of glycogen synthesis is the aptly named glycogen synthase enzyme. This enzyme takes our UDP glucose and links it to our glycogen chain. The mechanism for this is shown over here below. In the first step, we have our UDP glucose. This UDP over here is a really good leaving group, and it will actually leave by itself. This results in the formation of a half-chair intermediate, which actually mirrors in some ways the intermediate species found in glycogen phosphorylase in the other direction. This oxonium half-chair intermediate undergoes a nucleophilic attack by our target glycogen chain, which links our glucose to our glycogen chain, as shown over here below. Again, this chemical reaction links our UDP glucose to our growing glycogen chain, and it does so by leveraging the leaving group potential of our UDP glucose on our number 1 carbon of our glucose molecule, shown over here in green. This generates an unstable half-chair intermediate, which can then be attacked by our growing glycogen chain. The final step of our glycogen synthesis is the branching enzyme. What this basically does is it rebuilds our glycogen branches. Specifically, it takes a 7-glucose chunk of a glycogen chain and transplants it onto another glucose on our glycogen molecule. In other words, it takes a long chain and divides into 2 shorter chains, giving us more and more branches. The purpose for this is, again, to increase the number of ends or terminuses on our glycogen molecule, which allows for really rapid synthesis and breakdown depending on the conditions. Right? The more branches we have, the faster our glycogen can be either accessed or synthesized depending on the conditions. This does end the content for this particular class. At the end of the lecture, I do have some questions for you to look at. These are available in your template as well as on Moodle, and I'll give you a little bit of time to think about that and take them up in the next class. Also, in anticipation for next class, I would like you to read page 405 and 406 of your textbook. This will correlate to the first 2 pages of section 12.9. This section discusses the regulation of glycogen metabolism. When you're reading these pages, I would like you to keep in mind 2 things, or 2 questions. First, is shown over here. There are multiple stages involved in the regulation of glycogen phosphorylase. What are those stages? Maybe it might help to draw out a schematic or a map illustrating how the various regulatory factors play in or affect glycogen phosphorylase. Secondly, I'd like you to think about protein kinase A, or pKa. Does pKa enhance or suppress glycogen synthesis? Stepping further back and looking at glycolysis, what are some other pathways in which pKa is involved in? And is the role of pKa in glycogen synthesis consistent with its role in the regulation of other biochemical pathways, i.e. glycolysis?